Okay, let's read through lesson 2, the first 4 pages together. The first thing I want to look at is the reading guide's essential questions. So, what determines the expression of traits? So that's what we're going to be learning about. What's determining traits, and then how can we model inheritance? How can inheritance be modeled? And we're going to do that with some Punnett squares. We're going to show how we can model the pattern of inheritance. And then how do some patterns of inheritance differ from Mendel's model? So Mendel looked at traits that had just two factors, and one of them was dominant, one of them was recessive. Some patterns of inheritance do not follow that rule, and so we'll look a little bit at those. Let's look at the vocabulary next. Here are the words that we're going to be learning and using in this lesson: gene, allele, phenotype, genotype, and those are major points in this lesson, the difference between phenotype and genotype. Homozygous, remember homo, the prefix homo means same, so same. And then heterozygous means different, right? The prefix means different. So homo, same, hetero is different. Then we're going to be using Punnett squares again to model. And then we're going to be learning about, these are the things that, these last 3 are the ones that we were just talking about up here, these patterns that are different from the Mendelian model. So incomplete dominance, codominance, and polygenic inheritance. Okay, so this, to make the connection at the bottom here, says physical traits such as those shown in these eyes can vary widely from person to person. So they're different from person to person. What traits can you identify among them, and how do they differ? So looking at these eyes here, even the eyes that look like they're the same color, like these two are both brown, but this one's at the bottom is a bit clearer brown, almost coffee brown, and this one's almost black. You can see there's a difference in the darkness of the eye color, of the brown eye color. And this one sort of looks brown at first, and then the more you look at it, the more you can see some green in it. This one is, of course, blue, but there's a strong, on the outside, there's a ring around it. And then this one down here is blue, but there's not really much of a ring. It's more like solid blue. So even among the different eyes with the same color, there's going to be differences. All right, so let's see. From that, let's go on to the next page. I want you to look at the Launch Lab. If you have not done so already, you need to click on the link on this assignment in Google Classroom. You're going to be measuring the span of your hand, and you're going to, and please use metric, because we're scientists and we all use metric in science. Anyway, you're going to measure it in centimeters and then put your span, if you will, the width of your hand span. And then, so I want to take a look at that. So you are going to be doing that, and then we're going to kind of look at the patterns. The questions here, that think about this question, say, what range of hand span measurements did you observe? So remember, a range is the difference between the greatest and the least. So you're going to look at what is the difference between the greatest minus the least. So you'll look at that. And then number 2, do you think hand span is a simple Mendelian trait like pea plant flower color? And so I just want to think through this with you. These, I don't expect you to write these answers down anywhere, but it's something interesting to think about. So hand span, is it simple like a Mendelian trait? Well, remember that Mendel hypothesized that there were just two factors that made up each trait. And so he, for example, for flower color, he saw either purple or white. There wasn't anything else in between. There wasn't like a lavender where it blended. There wasn't like a little bit of purple and a little bit of white on each flower. It didn't work like that. But handspan is not going to, I'm hypothesizing that we're not going to find that people either have I don't know, a 35 centimeter handspan or a 20 centimeter handspan. I don't think we're going to find just two different values for handspan. So I'm going to hypothesize that handspan is something that has that is made up of many genes, and that it's not a simple Mendelian trait. But we'll look at that after you've put in your numbers for the Launch Lab. I will post the results of that next week, or you can go back and look at it yourself because you'll have access to that Google Sheets. All right, well, let's look at the text now. What controls traits? So Mendel concluded that two factors, one from each parent, control each trait. Mendel hypothesized that one factor came from the egg cell and one factor came from the sperm cell. What are those factors? How are they passed from parents to offspring? Well, they're passed through chromosomes, and we know this now, but Mendel did not have access to this information. He did not know. People had not discovered DNA and chromosomes, and they didn't have the technology that we have today to see that. So when other scientists studied the parts of a cell and combined Mendel's work with their work, these factors were more clearly understood. Scientists discovered that inside each cell is a nucleus that contains thread-like structures called chromosomes. Okay, so over time, scientists learned that chromosomes contain genetic information that controls traits. So this factor that we're talking about is the chromosomes. In fact, it's the genes on the chromosomes that control traits. So we now know Mendel's factors are part of chromosomes, and that part is genes. Okay, it's the genes on the chromosome, and that part of the chromosomes, and that each cell in offspring contains chromosomes from both parents. As shown in figure 5, these chromosomes exist as pairs, one chromosome from each parent. So let's take a look at that. This is called a karyotype, and we've talked about it before. Karyotype. Oops, T-Y-P-E. Okay, humans have 23 pairs of chromosomes. Each pair has one chromosome from the father and one chromosome from the mother. All right, so that means that these are the chromosomes, and remember, this is the way they look right before they separate in meiosis. Okay, so they're all scrunched up together. They're not spread out. They're all scrunched up together, and so this is what it would look like before they separate in either meiosis or mitosis. But one of these would be from mom, from dear old, and one from dear old dad, and that's the same for all of these right down to, we don't know which one is from mom, which one is from dad, but this one we do at the bottom. These are the sex chromosomes, so that's from mom because mom gave her the X, and then gave him the X, and this one's from dad because of course dad gave the Y to his son. So this is a male. Okay, all right, so we've looked at the karyotype, and these karyotypes, this karyotype is very, very common to look at. Let's see, let's just move on to the next page, shall we? Okay, so genes and alleles. Genes and alleles. Scientists have discovered that each chromosome can have information about hundreds or even thousands of traits. A gene is a section on a chromosome. So if you spread those chromosomes out, pull them, stretch them out, there's going to be a section of that chromosome that's like a recipe. That's a recipe for a particular protein. And that's what a gene is. It's just a recipe for a particular protein that's found on chromosomes. All right, so it has a genetic information for one trait. Remember that traits are really just an expression of the protein. Think about that for just a moment. Expression of a protein. For example, you have a darker skin color because your cells make a certain protein, the melanin in your cells that is made comes from this gene. The gene codes for the protein melanin. You got more genetic code for that, and so you have darker skin. So that's what we're talking about here. So for example, a gene of a pea plant might have information about flower color. Recall that an offspring inherits 2 genes or factors for each trait, one from each parent. The genes can be the same or different, such as purple or white for pea flower color. The different forms of a gene are called alleles. Pea plants can have 2 purple alleles, 2 white alleles, or one of each allele. We're going to be talking about that. They can be the same, and they can be purple. They can be the same, and they can be white, or they can be different alleles. And that's referring back to that same equals homozygous, different equals heterozygous. Okay, so in figure 6, the chromosome pair has information about 3 traits: flower position, pod shape, and stem length. How many alleles controlled flower color in Mendel's experiments? All right, so when we looking at flower color, 2 alleles controlled for the flower color. 2 different types of genetic code, if you will. All right, so let's look again now at genotype and phenotype. So look again at the photo at the beginning of this lesson. What human trait can you observe? Well, it's eye color, if you recall. You might observe that eye color can be shades of blue or brown. Geneticists call how a trait appears or is expressed as the trait's phenotype. What other phenotypes can you observe in the photo? Well, we observed, let's go back to the photo, because we observed not just eye color, but also you can see there's no like lip above the eye here, where there's a much larger one here. The difference between the kind of where the eye, I don't know, really know what that's called, but kind of like a lip of the eyes. And almost like this is probably an Asian eye, because they often don't have that. There's different amounts of eyebrows, the nice big eyelashes. I wish I had long eyelashes like that. Okay, so there are lots of different traits that we observed. Let's go back. Mendel concluded that the 2 alleles controlled the expression or phenotype of each trait. The 2 alleles that control the phenotype of a trait are called the trait's genotype. Although you cannot see an organism's genotype, you can make inferences about a genotype based on its phenotype. So for example, you've already learned that a pea plant with white flowers has 2 recessive alleles for that trait. These 2 alleles are its genotype. The white flower is its phenotype. All right, so take a look for a moment at these 2 chromosomes. So these 2 chromosomes are alike, and if you recall, we call these homologous chromosomes. Okay, and that just means that they're coding for the same type or the same gene, if you will, on the same location. So there's a location here, a loci here and here that's coding for the same gene. Now they may be coding for the same allele, or they may not. So here they're the same because it's axial flower, but here the bumpy pod and the smooth pod at the same place on these homologous chromosomes, they're both coding for the pod type, but one's coding for bumpy and one's coding for smooth. So these are two different alleles of the same trait. Okay. All right, let's move going down on to the next page, the last page we'll cover today. So symbols for genotypes. So scientists use symbols to represent the alleles in a genotype. In genetics, uppercase letters represent dominant. So actually, let's do this. So uppercase is dominant. And then lowercase represent recessive. Table 2 shows the possible genotypes for both round and wrinkled seed phenotypes. Notice that the dominant allele, if present, is written first. So for the phenotype round seed, the round seed could have either capital R, or capital R, small r, because it only needs one capital R to express the dominant trait. Okay, so it has either two of those alleles, or it has one allele and one that's masked, one recessive allele that you can't see because it's masked. Okay, but if it has a phenotype that is wrinkled, if it has the recessive phenotype, the wrinkled phenotype, then we know it must be homozygous recessive. That's the only way, because if the dominant allele was present, then it would express the dominant trait, but it's not expressing the dominant trait, so it must be little r. So a round seed can have two genotypes, big R, and big R, little r. Both genotypes have a round phenotype. So why does this heterozygous big R, little r result in round seeds? This is because the round allele is dominant to the wrinkled allele. But a wrinkled seed has the recessive genotype, little r. The wrinkled seed phenotype is possible only when the same two recessive alleles, little r, are present in the genotype. So you're only going to see the recessive trait in Mendelian genetics. You're only going to see this recessive trait present when it has homozygous recessive genotype. All right, so we use the word homozygous and heterozygous. I've been using that word. Let's just review it. So when the two alleles of a gene are the same, the genotype is homozygous. Both big R, so that's homozygous dominant, and little r, so that's homozygous recessive, are homozygous genotypes, as shown in Table 2. If the two alleles of a gene are different, its genotype is heterozygous, because different and hetero mean the same thing. So big R, little r is a heterozygous genotype. Here's our key concept. How do alleles determine the expression of traits? Well, alleles determine the expression of traits because if at least one of these two alleles is dominant, then the dominant trait will be expressed. But if both alleles are recessive, the recessive trait will be expressed. Okay, let's look at this mini lab. We are not going to do this mini lab. Instead, I'm going to have you do a Mendelian monster. So you're going to come up with a monster. So that's your next task, and we'll finish this reading this lesson later this week.